Welcome back to the Deep Dive. Today, we are taking on a disease that is just defined by its complexity. Multiple sclerosis, or MS.
Right.
For decades, we've known it's a neurological condition where the body attacks its own protective layer, the myelin in the central nervous system.
And we know it's multifactorial. It's not one thing. It's this nasty cocktail of risk factors. You need specific genetics. You need an infection with Epstein-Barr virus, environmental triggers.
And lifestyle.
And lifestyle, absolutely.
Well,
That combination has made it so hard to isolate what the actual trigger is. If you have 20 things in the mix, how do you prove one of them is the key?
it's almost impossible.
But the spotlight has really started to pivot to a pretty surprising player. The gut microbiome.
Right.
That whole ecosystem inside you isn't just digesting your lunch. It's playing a critical role in CNS inflammation.
And that is our mission for this deep dive.
Yeah.
We're looking at a truly groundbreaking piece of research, the MS twins study. And this wasn't just another study finding an association. This research gave us functional evidence, real proof, that specific gut bacteria can actually trigger an MS-like disease in a living animal. So we're going to unpack their incredible methodology, and we're absolutely going to name names.
We are the microbial culprits.
Okay, let's start there. With a disease this complex, with hundreds of gene variants involved, how do you even begin to design a study that cleanly isolates the gut bacteria?
Well, you can't. Not if you just compare a random MS patient to a random healthy person.
It's useless, right? Their genes are different, their diets are different.
Everything is different. It's all just noise. Too many confounding variables. The brilliance of this study was realizing they needed people who were genetically identical, but different in one key way.
And that's where the twins come in.
That's where the twins come in. They found an incredible group, 81 pairs of monozygotic twins, identical twins who were discordant for MS.
Meaning one twin had MS.
And the other twin was perfectly healthy.
That is just. It's the perfect scientific control. Why is having that identical genetic blueprint so important here?
Well, MS has this really strong link to the HLA complex, these immune genes. Unrelated people have totally different versions. But identical twins, you just wipe that entire variable off the board.
They start from the exact same genetic risk level.
Exactly. And
Right.
not just that. The sources point out that these twins grew up in the same house. They ate the same food, got the same childhood colds, often until they were, in their early 20s.
So if the genes are the same and the early environment is the same, then any major difference you find later in life has to be from something else.
It has to be. And their hypothesis was that the key difference was hiding in the gut, in their microbial signature.
They've basically taken a 200-piece puzzle and boiled it down to just a handful of pieces.
Which you can actually test. Now, the next challenge was how to sample them. And this is where the study goes from just being rigorous to being, I think, genuinely inventive.
Right, because they didn't just take fecal samples.
No, that's the low-hanging fruit. Feces just tells you what's leaving the body. If you want to find the trigger, you have to go to the scene of the crime.
So where's that?
The ileum. It's the final stretch of the small intestine. And the reason is the pathology of MS involves these rogue T cells. The ileum is where microbes and immune cells have their most direct, their most intimate, and sometimes hostile interaction.
Okay, break that down for us. What's in the ileum that makes it so critical for, triggering autoimmunity in the brain?
It's packed with something called gut-associated lymphoid tissue, or JOLT.
Jolty.
Think of GOLT as the central command for your gut's immune system. This is where specialized immune cells, specifically Th17 T cells, get trained and activated.
And Th17 cells are the pro-inflammatory ones, right? They're known to be major players in MS.
The key drivers. So if certain bacteria in the ileum start sending out inflammatory signals, the Th17 cells are right there, waiting. They're the first responders, and they get hyperactivated.
So the researchers didn't just take the easy route. They performed an enteroscopy, an invasive procedure, to collect microbial samples directly from the ileum of some of these twins.
Yes, with consent, of course. It shows incredible dedication. But that very specific site-specific sample, that's what made the next step possible.
The functional test.
The gold standard. They moved into gnotobiotic mouse models. They took the ileal microbiota from the twins and transferred them into germ-free transgenic mice.
Okay, let's pause on the mice for a second. The sources call them RR mice. What do germ-free and transgenic mean here?
Germ-free means the mice have absolutely no bacteria, nothing. Their gut is a sterile slate, a living test tube.
Wow.
And transgenic means they've been genetically engineered to be susceptible to an MS-like disease. It's called EAE. But here's the kicker. These mice only get sick if they're exposed to the right external trigger.
Meaning certain bacteria.
Certain bacteria.
That is just brilliant. So you take a genetically sensitive mouse, you give it the gut microbes from the MS twin, and then you give another mouse the microbes from the healthy twin.
And you see what happens. The mouse basically becomes a little biological indicator. You're testing if the microbial community itself is enough to flip the switch and cause disease.
And the results, they weren't fuzzy, were they?
Not at all. They were conclusive. The mice that received gut microbes from the MS-affected twins, they developed the MS-like disease, EAE, at a substantially higher rate.
Wow.
A statistically significant higher rate than the mice that got microbes from their healthy co-twin. That was the slam dunk.
In proof?
The proof that the microbial makeup alone, totally separate from the human's genes, can kick off CNS autoimmunity.
Did they see any other changes in the sick mice that sort of connected the gut back to the brain attack?
Oh, yeah. The mice that got EAE, they showed all the classic signs. They had a big increase in those reactive IL-17 producing T cells, the Th17 cells, right there in the gut tissue.
So the bacteria were definitely juicing up the T cells right at the source.
Directly at the goal T. And they also saw signs of mild intestinal inflammation, which suggests the bacteria were starting to damage the gut barrier itself.
And there was this other detail that was almost eerie. A parallel to the human disease related to sex.
Absolutely. When they looked closer, the disease was preferentially affecting the female mice.
Which mirrors MS in humans. It affects women 2 to 3 times more often than men.
Exactly. It suggests that whatever inflammatory stuff these bacteria are making, it interacts more strongly with female biology, just like it does in people. It just shows how relevant this model really is.
Okay, so we've established it works. Now for the big reveal. Who are the suspects? Which bacteria are carrying this pathogenic potential?
They zeroed in on 2 primary suspects, both from the Lactospiraceae family. The main culprits were named as Eisenbergiella tae, or just E-tai, and its accomplice, a bacterium called Lachnoclostridium.
E-tai. Okay, so were these bacteria just like everywhere in the samples from the MS twins? Were they super dominant?
And this is one of the most surprising parts of the study. No, not at all. They were often just minor players in the original samples, but when you put them into the mouse, these 2 species showed this powerful blooming, a rapid expansion.
So they took over.
They took over. It suggests that while they might be kept in check in a normal gut, under certain conditions, maybe like a Western diet, these minor species suddenly get an advantage and can run wild driving the disease.
That's a critical point.
Yeah.
It's not about just having the bacteria. It's about the environment that lets them switch their role.
Exactly.
Well, let's follow that thread with E-tai. Its mechanism is like a real Jekyll and Hyde story. What's its normal, helpful job?
Normally, E-tai is a good citizen. It's a fiber fermenter. You eat fiber, it breaks it down, and it gives you back these amazing things called short-chain fatty acids like butyrate.
Which are anti-inflammatory and great for gut health.
The best.
But the research describes this total metabolic flip. What's the trigger that turns it into a, quote, mucus-eating machine?
The trigger is when its favorite food fiber is gone, but there's plenty of simple sugar around. When the fiber isn't there, E-tai gets hungry. It has to find another source of carbon.
And that source is us.
It's our own protective mucus layer that lines the intestine. It literally starts eating our gut lining to survive.
So it's punching holes in the intestinal wall. That sounds exactly like what people call a leaky gut.
That's the descriptive term for it, yeah. Increased intestinal permeability. And by degrading that mucus layer, the physical barrier between everything in your gut and your immune system, the LT is breached.
And that alone is highly inflammatory.
Highly. But the damage isn't just physical. While it's eating the mucus, it's metabolically stressed. And the waste products it starts pumping out. That's the real inflammatory fuel.
What kind of waste products?
When it's stressed, its metabolism shifts and it starts releasing ethanol and succinate. The source material had this really memorable term for them.
What was it?
Petrofumes.
Wow, petrol fumes.
Which is perfect, right? These compounds are signals that directly stimulate the immune cells waiting right there on the other side of that damaged wall.
So you have a leaky barrier, and you have these petrol fumes pouring through. What's the final domino to fall that leads to the attack on the brain?
Those fumes super activate the Th17 T cells in the galt. They become extremely aggressive. They leave the gut, enter the bloodstream, and now they're primed to get across the blood-brain barrier.
And once they're in the central nervous system,
They launch their attack, mistaking the myelin sheath for an invader. And that's your MS attack, a whole cascade fueled by bacterial stress.
And what about the accomplice, Lachnoclostridium? What's its role in all this?
Lachnoclostridium is the enabler. It helps create the inflammatory environment by crowding out the good bacteria, the ones that produce anti-inflammatory compounds.
So it suppresses the good guys.
Right. It makes sure that this microbial imbalance, this dysbiosis, can continue, which lets E. tae keep doing its damage.
Okay, this is an incredible finding from a mouse model. But what does this mean for you listening right now? How does this connect to the real world?
It connects directly, and I mean directly, to the global data on MS rates. You look at the places with the highest rates of MS North America, Canada, Western Europe, Australia.
What do they all have in common?
They have some of the lowest intakes of dietary fiber in the world.
The Western diet.
The Western diet. Again, most people in those regions are eating, 12, 15 grams of fiber a day? That's half the WHO recommendation of 30 grams.
So it really paints a picture where this low-fiber, high-sugar diet is systematically starving the good bacteria.
And it's starving E. tae of its preferred food. That forces it to turn pathogenic and, potentially increases the risk of this disease.
The knowledge here is so powerful because it gives you immediate, tangible action. If your diet is the switch that turns E. tae into a mucus eater,
Then changing your diet is how you flip that switch back.
right?
The main takeaway is to intentionally feed your microbes. Feed them fiber. When you eat lots of different fibers, from legumes, nuts, whole grains, you keep your good bacteria happy, and you keep guys like E. tae busy and peaceful.
And they reward you. They give you butyrate and other good stuff that calms the immune system down.
Instead of ethanol and succinate, you're moving away from vague advice like eat healthy to a very specific goal. Use fiber to make sure your gut bacteria are making good things, not inflammatory things.
And that has to lead to therapeutic potential.
Oh, absolutely. If this disease facilitation is really restricted to just these two specific organisms, that opens up totally new, non-invasive strategies.
Like what?
Well, instead of broad immunosuppressants, imagine a targeted probiotic that only suppresses E. tae, or a specific prebiotic fiber that only feeds the good bacteria so they can outcompete the bad guys.
That completely changes the paradigm. You're not just treating the damage after it happens. You're proactively changing the environment to stop the trigger in the first place.
We functionally identified the specific triggers, E. tae and Lachnoclostridium, in the most critical site, the ileum. I mean, this whole deep dive, from the twin cohort to the sampling to the mouse model, it's just a monumental step forward for understanding the gut-brain axis.
So here's the final thought we want to leave you with. MS development requires so many things to go wrong. The right genes, an EBV infection, your environment. Given all that complexity, how significant is it that an imbalance in just two species of bacteria, often just minor players, could have the power to functionally tip that entire scale towards CNS autoimmunity?
It's massive. It means that while you can't change your genes and you can't change whether you got EBV as a kid, you hold this immediate daily power over one of the most critical risk factors. Your fiber intake isn't just about digestion. It's a daily decision that influences your entire immune system. It's a powerful thought to mull over next time you're deciding what to eat. You're either feeding an inflammatory fire or you're strengthening the walls that protect you.